Hello all, myself Shruthi. Welcome back to the videos of Bio8 solution. So here I would like to explain and introduce one batch of students who are from Garden City University, wherein they have worked on isolation and identification of biosurfactant from garbage soil through molecular techniques. So this field is growing field wherein biosurfactants can serve as green alternative in different areas due to their ecological acceptance as they are biodegradable and non-toxic in nature. Nowadays, biosurfactants are predominantly used in pharmaceutical oil industry and for the bioremediation of pollutants. So here, if we see what are biosurfactants, these are the surfactants of microbial origin. So basically, biosurfactants are amphiphilic compounds. They are secreted extracellularly by microorganisms, and biosurfactants contain hydrophobic and hydrophilic moieties that reduce the surface tension and interfacial tension by accumulating at the interface of hydrophobic substances, and thus increase the solubility, bioavailability, and subsequent biodegradation of the hydrophobic or insoluble organic compounds. So they are synthesized extracellularly, and many microbes are known to produce biosurfactants naturally in large relative quantities. So these biosurfactants can basically used in oil recovery from the rock surfaces and porous surfaces, and the removal of heavy metal from contaminated soil, treatment of rough skin with biosurfactant as a moisturizer. It has got lots of applications, so it can enhance the emulsification of hydrocarbons. Thus they have the potential to solubilize hydrocarbon contaminants and increase their availability for microbial degradation. These compounds can also be used in enhanced oil recovery and may be considered for other potential applications in environmental protection. So if we see, these biosurfactants can be produced from so many biological materials like bacteria, fungi, and applications is seen in the pharmaceutical industries, in bioremediation, in oil recovery processes, in cleansing, cosmetics, all these case. So nowadays it is gaining lots of interest. So with view of this, our students collected the sample from different garbage dumping yards. They cultured and isolated the pure bacterial colonies from the soil, and then the morphological and microscopic analysis of the organisms were done. Then the organisms were identified and characterized by doing Sanger sequencing method, and later the organisms which were identified was checked for the property of biosurfactant by doing CTAB assay and penetration assay. So if we see the flow of the work, they have collected the garbage soil from the plastic dumped areas, and from there they isolated the pure colonies, and then the pure colonies were subjected for gram staining to identify whether they are gram positive or gram negative. So they were able to identify they belong to Aeromonas and Pseudomonas species, and further from these organisms DNA was isolated, and from the genomic DNA 16S rRNA gene was amplified and further subjected for Sanger sequencing. So from this, with a sequence of 16S rRNA, we were able to identify the species as Aeromonas cavea and Pseudomonas originosa, showing maximum percentage similarity. So after the isolation of the microorganisms, they were checked for the production or the biosurfactant property of the organisms by doing two assays. They are CTAB agar plate and penetration assay. In case of CTAB agar plate, these organisms were plated on the mineral salt medium containing CTAB and the basic dye methylene blue. So based on the degradation of the dye and formation of the ring around the agar well, we were able to tell whether the organism can produce or behave as biosurfactant or not. In the same way, in case of penetration assay, hydrophobic paste consisting of oil and silica gel was prepared, and 150 microliter of this paste was added to each well of microtiter plates. Above this, 90 microliter of this culture supernatant and 10 microliter of saffronin stain was put. So if the supernatant contains biosurfactant, it will cross the oil barrier and enters the paste. So this is how we were able to identify whether the organisms could be a good biosurfactant. In case of penetration assay, it was seen that the upper phase changed from red to cloudy white, whereas in case of CTAB assay, there was a blue ring formation around the cultures. So with this we could say that these Aeromonas and Pseudomonas were the two species which were isolated and characterized from garbage soil. They have the capacity to behave as a biosurfactant. So in the current era of biotechnology, these biosurfactant producers can be utilized in bioremediation technologies, and so that it will be helpful for various industrial processes. And also this biosurfactants, it acts as a multi-purpose products, hence it is accepted worldwide as suitable, sustainable, greener alternatives. So we should come up with innovative formulas to isolate this biosurfactant from various sources and how it can be applied to the industry. So these are called as the chemical specialties of the future. So here we will see how the students have understood the project, how they got interest in picking up this project, and how they have performed the project, and what was the solution outcome what they got from the project, and how satisfied they are with carrying out the project at BioEdge Solutions. Thank you.
Greetings to all. Myself and my fellow friends are going to do a small video on our project Isolation and Identification of Biosurfactant Bacteria from Garbage Soil through Molecular Techniques. Our main goal of this project was to isolate the bacteria and examine whether they are having a biosurfactant property or not, and also if they had, to examine the ancestral relationship between those two bacteria. As mentioned above, we isolated the bacteria, we performed biochemical as well as the molecular tests, and through the bioinformatic tools we obtained the data. Biosurfactant bacteria are not only important to our environment, they are also having many applications. Like they are having industrial applications in food, cosmetic, petrochemical, etc. They are very important to our environment and very much needed to our environment. They are not only having applications in the industries, they also play a very vital role in oil spill control and also in biodegradation, detoxification, and many more. As I said, there are many different species of bacteria which are found in the soil which degrade the plastic and show the property of biosurfactants. So we need to examine all those bacteria and record the data so that it will be useful to the environment and also to us to examine the bacterial species.
Hello everyone, I am Sharanjaba. I am here to quickly tell you all about the materials and the methods. So the first step is the collection of the soil samples. 2 soil samples were collected from 2 different regions in the garbage dumping yards, followed by which comes the culturing of the bacteria. So in culturing of the bacteria, first step is the serial dilution, followed by which the LP agar plates were prepared, streaking was done, and pure colonies were obtained. The third step is the morphological and the microscopic analysis. In this, the gram staining method was performed, after which comes the main step, that is the DNA isolation, in which the DNA silica column method was done. After the DNA isolation, gel electrophoresis was carried out. After gel electrophoresis, PCR was done, and in PCR the primers which were used are 27F and 1492R. So after the PCR, gel elution was done, and after gel elution, followed by which Sanger sequencing was done. So in Sanger sequencing, PCR processing, capillary electrophoresis and electropherogram was put into action, after which data analysis was performed. So in data analysis was done, and which the final step is it was checked for the biosurfactant property, under which two methods were carried out. The first one is the CTAP agar plate, and the final was the penetration assay.
For the experiment, identification of biosurfactant from garbage soil through molecular technique. Initially, we collected the garbage soil from two different places because a comparison will be made between two samples, and the bacteria were cultured and various biochemical tests were done to identify the microbes present in it. Gram staining were done to distinguish and classify bacterial species into two groups, gram-positive bacteria and gram-negative bacteria. When we did the gram staining, the result obtained was pink color, which indicate the presence of gram-negative bacteria. Here we use CTAP assay to find the presence of biosurfactant. Blue color formation was found around the colonies in the petri dish. To confirm the presence of biosurfactant property, the experiment penetration assay were carried on and the result was positive. DNA was isolated using DNA silica column method. DNA strands were obtained and was visible in the vial. DNA strands which was isolated was run in gel electrophoresis for the purity and concentration. So the result obtained was compared with 1 kb ladder. After all these experiments were conducted, Sanger sequencing of the sample was done and the phylogenetic tree for the same was obtained. It enabled us to understand about the nature and properties of the gram-negative bacteria we were.
In the present world, it is important to explore new solutions and choices for the problems and situations that would come up in our way. Working in a scientific field always provides us with an opportunity to work for the society. For the past few years, it has been very evident that majority of human activities has had a negative impact in our nature. So the project undertaken by my group was isolation and identification of biosurfactants from garbage soil through molecular techniques. So working on this project, we mainly focused on whether or not this property of bacteria could be put into practice in an efficient way so that it would benefit our nature. With the increasing industrialization, there has been increase in the depletion of natural resources as well. So, using this property of bacteria and putting them into practice in an efficient manner would help the nature to heal, as well as it would also help to build an economically independent society and a healthy society. The work and experimentations on this may take a long time, but it will be worth it. It may only have a small impact, but it would mean something to the society. So we should always focus on working on such projects which would prove beneficial to the society in all manners. Working in this project has helped us to learn a lot as science students, and it has only encouraged us to learn more and more about such things and encouraged us to take up more such projects in the future that proves beneficial to the society. So we would like to thank BioEdge for providing us with an opportunity to work on such advantageous and interesting project. Thank you.